Hi, everyone, and welcome. Again, this is our OTS webinar series, Trainee Spotlight today. My name is Erin, and we also have Hassan, who will be your OTS co-host for this webinar today. And we also have two excellent trainees who will share their research. First, we will have Priyanka Shukla from SRI Biosciences, and then we will have Remko Goossens from the Leiden University Medical Center. So we encourage you to ask questions through the Q&A feature. You can just pop them in there, and at the end of each trainee's talk, we will coordinate the Q&A session. So thank you for attending. We're really looking forward to having these speakers. And Priyanka, do you want to take it away?
Thank you so much for the nice introduction. And thank you, OTS, for giving me this opportunity to present this current ongoing work. But before we dive in, I just want to give credit to my mentors, Drs. Kathleen Brown and Michael McGuire, for their immense support and help in building up this story. Today, I have the pleasure to discuss with you molecular guidance systems for intracellular target delivery of the siRNA bioconjugates. siRNAs have immense therapeutic potential for the treatment of gene-related diseases ranging from cancer to neuropathy to autoimmune diseases. However, their bench-to-bedside translation in recent years has faced several challenges in targeted intracellular delivery to non-hepatic tissues. The widely used lipid-based methods for the delivery of nucleic acids have been associated with ineffective delivery to the target sites outside of the liver. Poor pharmacokinetics that leads towards several off-target effects involving cellular toxicity and issues with tissue specificity. Thus, there is an urgent need to develop a better intracellular delivery technology platform that can deliver any type of biotherapeutic cargo with high cell specificity and affinity to the target cells. Here at SRI Biosciences, we have developed FOX3 molecular guidance system technology that can rapidly, effectively, and specifically deliver the therapeutic cargo to any specific cell type. FOX3 is a high-throughput selection platform that identifies peptides in 6 to 12 weeks to target specific cells. These peptidic MGSs bind to the target cell and undergo rapid cellular internalization through receptor-mediated endocytosis to deliver the therapeutic cargo to the target cells. This robust technology overall focuses on increased delivery efficiency to the desired cells and tissues. Thanks to this technology, we now can deliver various payloads, from proteins to biotherapeutic small molecules, and in our case, nucleic acids, to the correct location within the target cells. Most importantly, they do not internalize into the non-target cells. Interestingly, MGS drives the targeting, not the payload. As I mentioned previously, MGSs achieve rapid internalization with high cell specificity and affinity. Lastly, cargo is incorporated into the MGS scaffold without disrupting the cell binding and the internalization of the MGS. We have a panel of over 50 MGSs for different cell types. However, it is important to note that we can generate MGSs to target any specific cell types and for any therapeutic application. MGS selection has been performed on primary cells as well as cell lines. Next, there are several advantages of our MGSs over other drug delivery approaches. For example, antibodies can deliver any therapeutic payload to target cell types, but are rarely internalized into the cells, thus preventing their intracellular delivery. Cell-penetrating peptides, on the other hand, are short peptides that have been used to transport a wide variety of cargo, but their uptake mechanism is still the most challenging question. CPPs are known also for their non-specificity and also possess poor affinity. In short, MGSs are highly cell-specific and are internalized in large quantities within the target cell. They can be rapidly generated in 6 to 12 weeks for any cell type and can be chemically synthesized and conjugated to any drug of interest with no loss in the internalization capability. I would like to highlight a little bit about our two lead MGS candidates from our FOX3 platform that specifically target lung cancer cells. And they are MGS1 and MGS2, which are also the lead candidates in this current study. These two MGSs have been optimized and assessed for their behavior in vivo as well as in vitro. As shown in this figure, we set up subcutaneous tumors in the flank of an immunocompromised mouse. MGS1, Elexaflor 647, was administered through tail vein IV and allowed to circulate for 24 hours. MGS1 was able to accumulate in the H2009 tumor mass on the animal's right flank. A tumor formed from H460 cells, which do not express the receptor for MGS1, is present on the left flank. As you can see, the H2009 tumor accumulated MGS1, while the H460 tumor did not. We performed similar experiments with other MGS, and that is MGS2, which accumulated in the target H1299 tumor, but not in the H460 tumors. Thus, we can confidently say that our MGSs show exquisite cell specificity and perform as expected in vivo. The MGS6 accumulates in the target H460 tumor, but did not show in the control H2009 or H1299 tumors. We next conjugated our MGSs to our model gene GAPDH. The conjugation of the MGS was done to the passenger strand of the siRNA. PEG spacer separated the MGS from the antisense strand of the siRNA, thus minimizing any effect of the MGS on the antisense strand. We then moved forward to assess the peptide-facilitated siRNA uptake using quantitative flow cytometry. As I mentioned, using GAPDH as our model gene, we conjugated the MGSs to the passenger strand of the corresponding siRNA. Elexaflor 647 was incorporated into the MGS scaffold to allow us for biological assessment of the cellular uptake. The input concentration for the treatment all across the board was 25 nanomolar, and the target cell lines were kept under the treatment for 1 hour. As you can see, MGS1 and MGS2 enables the intracellular delivery of the siRNA to the targeted non-small lung cancer cell line. MGS1 siRNA, as shown on your panel to the left, and the MGS2 siRNA conjugates, shown on the panel to your right, are internalized into the target cells at significant levels. We did not see any internalization with the cells only and the siRNA only treated samples, indicating that the conjugation with the MGS does not affect the delivery of the siRNA inside the cells. And also, The attachment of the MGS to this highly negatively charged cargo did not impact the delivery capabilities of the MGS. We further validated our results using confocal microscopy, as shown here in this figure. The MGS1 siRNA conjugate is internalized over time in the target H2009 cells. The input concentration again was 25 nanomolars. Cells only and the siRNA treated only samples served as controls. The nuclear stain here is hooked. The green stain here is wheat germ agglutinin labeled to Alexaflor 488 that marks the plasma membrane. Red staining indicated here in MGS1 treated cells indicated the cellular uptake of the peptide and the siRNA conjugate. Also, the MGS1 siRNA conjugate is efficiently internalized and continues to accumulate in H2009 cells over time. We saw the similar results with the other MGS, and that was MGS2 siRNA conjugate in the target cell line H1299. The MGS2 siRNA conjugate efficiently internalized in the target cells. Again, the red staining in the MGS2 treated cells indicated the cellular uptake of the peptide and the siRNA conjugate, showing that the MGS2 can internalize into the cells and the siRNA retains its activity. Also showing that the MGS drives the internalization, not the cargo. We then moved forward to assess the knockdown efficiency of the MGS-delivered siRNA using both our MGSs, MGS1 and MGS2 in the target cell line. Our input concentration here was 25 nanomolar for the treatment. We kept the treatment on the respective cells for 72 hours. At the end of the 72 hours, we harvested the cells and processed them for downstream analysis. As seen here, we saw effective knockdown by both the MGSs, which was more than 60%. The MGS1 and the MGS2 delivered siRNA did enable the robust gene knockdown. We did not see much impact from the untreated and the MGS treated alone samples. Thus, MGS directly facilitated the intracellular delivery of the siRNA without the requirement of any transfection reagent. And the siRNA retained its functional efficacy. Interestingly, we did not see any effective knockdown in H460 cells, which are our control cell line. The reason is these cells lack the MGS receptor and thus were not able to internalize the MGS siRNA conjugate. Experiments were performed in the same manner as discussed previously. We did not see any impact on the reference gene PPIV used in these experiments, and all the experiments were normalized to no treatment. We compared our effective delivery of the siRNA via our MGSs with the commercially available Dermafect reagent, which served as a positive control. The targeted intracellular delivery using MGSs did in fact retain the functional efficacy of the siRNA, resulting in low IC50 without involving any lipid carrier. This clearly defines the impact of this enabling technology on MGS-delivered siRNA silencing ability. We also witnessed that the IC50 is comparable to the EC50 measured to peptide binding and internalization. These results were obtained through quantitative real-time qPCR. Thus, we can say that the MGS-delivered siRNA is effective in showing robust mRNA knockdown and is concentration dependent. We also looked at the effect of the MGSs and the MGS-delivered siRNA conjugate on apoptosis. As shown here in this figure, we did not see much effect on the cytotoxicity on H1299 cells in the lower panel by MGS2 delivered siRNA. With MGS1 delivered siRNA in the panel, in the top panel, in the H2009 cells, we did see some effect that we assume appears to be due to the MGS, not by the delivered siRNA. This toxicity that we see in case of MGS1 is likely to be less effective in vivo. However, when compared to the other standard delivery moieties, we still see less toxicity and higher viability. This brings me to summarize these results. Thus, the conjugation of the siRNA payload to the MGS does not alter the cell-specific uptake of the peptide. MGS siRNA conjugates are able to suppress the gene expression of the target gene of interest without requiring any lipid carrier. Our work clearly demonstrates the effective targeted intracellular cell-specific delivery of the functional siRNA. If you have any questions, suggestions, or if you would like to know more about our FOX3 platform, then please free to contact Melissa Wagner and Intron. With that, I would like to thank you for your time, and it was great to be a part of this webinar series. Thank you.
Thank you so much, Priyanka. That was excellent and very exciting work. We're gonna wait for some questions to come in the Q&A, but I'll start with some questions actually for you. One of the first questions is, what is the yield of the conjugation? Is it like, do you get like, is it a one-to-one conjugation yield? Do you have any difficulties, putting the MGS on the siRNA?
Actually, because of the ease of the click chemistry, we do not see any issues so far.
Okay.
So the yield is always perfect.
Okay.
And yeah, we have done some serum stabilities and all that things to make sure that the conjugate stays intact. So we do have those data available.
Okay, and we have some questions from the chat. So question is, how is the endosomal escape ratio?
So we are looking into that, but as I showed you, the amount of molecules that are getting internalized, we assume that even if the 2% of them escape out, still we have a lot out there to effectively do the endosomal escape.
And we have another question in the chat. They're asking, can the MGS be nebulized for inhalation?
So can you repeat that?
The question is, can your molecules, the MGS, be nebulized? So can they be made into aerosol for inhalation delivery through the mouth or the nose?
I think yes, but I think this question should be addressed to the chemist.
Okay.
Yeah, I'm sorry, I don't know much about. but I believe so that they can be.
Yeah, and if you said you guys did stability measurements,
Yes,
that
we have, so as I mentioned, that we have over 50, more than 50 MGSs that we have validated for various cell types. And of course, they go through a rigorous selection process and also the validation process. So for these two lead MGSs, which are MGS1 and MGS2, we have done serum stability test, and for up to 24 hours in human serum, they remain intact more than 75%. So again, as I said, we have all these data. So if you have a curiosity about this platform and want to know more about it, then please, we will be more than happy to answer any questions. So reach out to Melissa.
Okay. We have another question, and I'm not sure if you can share this, but Bruce is asking, what receptor are you targeting for uptake, specifically for the data that you've shown?
Yes, we do encounter this question very often. Very soon, I vouch for it. We will have this answer for all the audience, but for the moment, we know the receptors for these MGSs, but again, not in the liberty to discuss it.
Mm-hmm. Okay. Another question is, I guess maybe you touched upon this. Romana is asking, has the in vivo efficacy of the conjugates been evaluated?
So I'm in the process of accumulating those data, but that's why I showed our previous results that these MGSs are very specific for the target cell types on which they were panned and discovered. So if the MGSs are going and accumulating in the target tumor mass, and if I'm conjugating the siRNA to it, I firmly believe that we are going to see an effective delivery and an effective gene knockdown. So again, very soon I will have those answers for you ready.
Excellent. And Mark is asking, can these MGS or compounds be shipped at room temperature? So basically, what is the storage recommendations? Are they stable at room temperature, or do they need to be frozen or put in, 4 degrees?
Sure. So yes, once they are conjugated to the siRNA, we like to keep them frozen because we have a conjugate attached to it, but all throughout the processing time, like when they are being synthesized and when are they being conjugated, they are going through the process at room temp. So yeah, they are pretty stable.
Okay, and I guess a question from me is, I don't know if you can disclose this, but I'm assuming these are also made from amino acids, or like they're peptides in their nature. So how do they differ from the comparison that you made to cell penetrating peptides? Is it like your platform, the ability to find the targets, or are they made from like additional components?
So the peptides identified using the phage display are very, very specific towards that target cell types on which we are testing them. And that itself is a remarkable property of this technology that the cell penetrating peptides are not effective of. We are not seeing any off-target effects when I talk about the siRNA conjugated prep, but also once these peptides are basically specifically targeted for the target cell type. So they can differentiate in the milieu between the diseased cell and the healthy cells. So a cancer peptide is very specific for cancer cells. A peptide that we have, for example, for microglia is not going to bind to the cancer cells. So for us, cellular affinity and cellular specificity is very, very important. And that's why I say that this is a robust platform that is very specific for intracellular delivery of any payloads.
Awesome. Thank you so much, Priyanka.
You're very welcome.
Great. So we're going to move to our next speaker now. So just a reminder, this is Remko Goossens from the Leiden University Medical Center. So Priyanka, if you just want to stop screen sharing, and then Remko can get his presentation up. Take it away.
Thank you, Erin. So indeed, my name is Remko Goossens. I'm a postdoc in the group of Annemieke Aartsma-Rus in the Department of Human Genetics in Leiden University Medical Center in the Netherlands. And this project that I'm presenting to you is a collaboration with Sarepta Therapeutics. And the baseline is that we are looking into how AON-mediated exon skipping can be further improved for targeting exons in Duchenne muscular dystrophy. So a little bit of an introduction on Duchenne and the DMD gene. So the dystrophin gene is the largest gene in the human genome at 2.2 megabases, and it contains 79 exons. Only 0.7% of DMD is actually protein coding. So you can imagine that there is extensive splicing going on when this transcript is being produced in the cells. Introns range between 107 base pairs and up to 250 kilobases, but are on average 28 kb. And to complete transcription of the entire molecule takes estimated about 16 hours. So in Duchenne muscular dystrophy, this dystrophin protein, which is normally in panel A here, is disrupted by internal deletions very often, or nonsense mutations. And then, of course, you cannot have this entire protein, and this will lead to muscle damage over time. And this is very severe muscle disease, and patients become often wheelchair dependent in their second decade of life, and very often pass away in the third or fourth decade of their life. I've put an quite extensive introduction because I've noticed from previous presentations that to understand this project properly, it's necessary to give the entire story. So back in 2016, a postdoc in our lab, Isabella Cazzoli, published a paper where she was looking at splicing in multiple steps, and then specifically co-transcriptional splicing of the DMD gene. So classically, splicing is thought of as single-step splicing, where the intron is just taken away as a lariat, and then you have your mature exons joined together. But what actually happens is that multi-step splicing occurs in about 22% of human introns, and this can consist of recursive splicing, so that the intron is removed from the 5' or 3' end in multiple steps, or nested splicing, where first a middle part of the intron is removed before the transcript is further matured. What she did to figure this out was to isolate pre-mRNA from myotube culture. So myotubes are mature muscle cells which express the dystrophin protein, and also, of course, the mRNA. She did cDNA synthesis and sonogated it to shear it into small pieces, and then she captured the entire DMD transcript, including introns and exons, with biotinylated probes. And these probes were first validated to have no sequence bias or enrichment bias. So, and after high sequencing and analysis on the custom pipeline that they developed themselves for non-sequential and recursive splicing, they noted where recursive and nested splicing was going on. But what they could also do is make a map of the coverage, which was normalized for the read maps. So which is displayed here, and to walk you through it, what it means is introns which are retained longer in the transcript will have a higher coverage. So for example, here exon 40 has a really high coverage, which means it was excessively present in the data set. And this is not related to intron length, because as you can see, this bar here, or this line here, is the intron length in a log scale. And you can see that there doesn't seem to be a difference between coverage or the intron length. This allowed Isabella to make an intron map of non-sequential DMD splicing, which is depicted here. And what you see is that, for example, the introns which are marked in red are retained longer than the introns which are marked in green here, which are also, for visualization, the exons are joined together. And what this means is that, for example, if you have a transcript, it could be that you still have intron 2 present here, while intron 3 or intron 4 have already been spliced out. And that we call non-sequential splicing of the DMD mRNA. And we say that these are so-called slow introns and these are fast introns. Well, almost at the data part. And then the final thing we should note is that for DMD, there are a few therapies already in existence. For example, one is eteplirsen, which is an exon 51 skipping PMO AON. And the idea is that if you have a diseased allele, as here, depicted here with exon 50 missing, then you have a disturbed reading frame and your protein will not be produced properly. But now if you use an AON to mask exon 51, exon 49 will be spliced to 52, and this will restore the reading frame, and then you should get a slightly shorter but functional dystrophin molecule. Currently, there are 4 different PMO AONs approved for Duchenne, 2 for exon 53, 1 for exon 51, and 1 for exon 45, but clinical efficacy and patient benefit can be improved further. And also part of this project is to see which new exons could be favorable targets to develop AONs first. So, to investigate the relationship between splicing speed and skippability, we assume that exons which are flanked by a slow intron at either the 5' or 3' end will be more susceptible to exon skipping, simply because if the intron is retained longer in the mature transcript, your AON will have a longer opportunity to bind and disrupt the splicing machinery. So for this purpose, we selected these exons here as targets for our approach. For example, here exon 17, which is flanked by a slow intron at both its 5' and 3' end, and then, for example, exon 55 here, which has a fast intron on both ends. And then also we chose, for example, exon 50, which is flanked on its 5' by slow and on its 3' by fast intron. Then the hypothesis is, will the fast intron or the slow intron skip better? So we have a little race, we have our little exon, we cover it in PMOs, and then we're going to figure out what is actually the better skipper. For the PMO design, we had some requirements. I'll briefly go through this. All oligos had to be a 24 or 25 mer. All exons were chosen to be out of frame. So this let us include 38 out of the 79 DMD exons. We designed almost 10,000 unique PMOs by tiling the full sequence with this design properties. And then we wanted to have the AONs to be as chemically similar as possible. So we matched the GC content and the TM, and we excluded PMOs that had an unfavorable TM or an unfavorable GC content. So that means lower or higher than 40 and 60% respectively. Then to make sure that we do not have PMOs on each side of the spectrum, so we are comparing a very low melt temperature with a very high melt temperature. All PMOs outside one standard deviation of either TM or GC were excluded. And then after that, self-dimerization potential was calculated, and all PMOs with unfavorable predicted free energy were excluded as well. So then finally, the remaining PMOs were chosen to form an equal distribution along the 5' middle and 3' ends. And to make up for the differences in exon length, we made sure that there was a normalized distribution by dividing the length of the exon by 30. So you can visualize it as this, as a visual example. Again, exon 17 of 176 base pairs. We first designed all the PMOs, which are shown here. After removing the PMOs with unfavorable GC and melt temperature, we exclude already quite some. Then the standard deviation removes more, and then after the predicted free energy, you can see that again we drop out a bunch of PMOs which are highly likely to either bind to themselves or make a hairpin. And then after that, we manually selected the final distribution to be neatly covered over the rest of the exon here. So that gives us 6 PMOs for this one exon. Well, and then a long story short, if we then electroporated our PMOs into the muscle cells and assess the exon skipping potential by using a femtopulse system, which is similar to the bioanalyzer for the people that use that, you can assess the skipping efficiency of these PMOs in all the different exonic regions. And then what you see here, for example, is the wild type, again for exon 17, I'm exemplifying. And then you can see that we have, as farther we go, then we see a lower efficiency here of the different PMOs. And for example, here where we have a fast, slow intron flanking exon 65, you can see that there's barely any exon skipping occurring at all on a scale of 1 to 100%. To condense this data a bit, I grouped all the 3 different exons corresponding to each class in this graph. And then you can see that if we have the slow, so 5' slow, 3' slow exons here in orange, or the slow, fast exons, then you can see that the skipping efficiency in general is much higher than when a fast intron is flanking the 5' end of an exon to be skipped. This is further exemplified here by condensing the data further. If we just like base the grouping on either the 5' or the 3' flanking intron, we can see that there's a significant difference, but of course there's always exceptions. That slow 5' is easier to skip than a fast 5'. On the other hand, it doesn't matter how fast the 3' end is processed, either slow or fast, the skipping efficiency doesn't seem to be different. So for this part, exons with a slow 5' intron are easier to skip. So the hair is definitely in this case not the winner. Then, because we already had all this data and all these PMOs, we wanted to investigate the dynamics of the skipping efficiency a bit further. So to assess the optimal targeting position in the exon, we wanted to validate whether targeting the 5' or 3' end of an exon flanked by either slow 5' and fast 3' or fast slow introns is more efficient. So basically, we wanted to see if it still mattered if we would target closer to the fast or closer to the slow intron. And that is summarized on this graph. So what you have on the y-axis is the exon skipping efficiency over wild type from 0 to 100%, and then the relative binding position of each AON in the exon on the x-axis. And what you can see is these colored lines for each exon are negative, which means that if you have the 5' binding ASOs, they skip quite efficiently. But if you go to the 3' binding ASOs, they are not efficient at all. So this confirms a hypothesis which was already present in the field, but was, as far as we know, never routinely checked by using such a large array of different AONs. Then we also wanted to see whether the detrimental effects of skipping the transcript out of frame were an important factor in our model system, because what we did is we skipped all these exons in a wild type cell line. And because we were using an out of frame selection of exons, you will actually cause nonsense mediate decay potentially. So what we did here is when we have exon 51 or exon 53 skipping, for example, we see in our wild type cell line, we see relatively low exon skipping levels. But then we used 2 cell lines which are either amenable for exon 51 skipping or exon 53 skipping. So these are Duchenne patient derived cell lines. And then we see that exon skipping levels are much improved. But importantly, what we do see is that the relative contribution is similar. So for example, here, the first BMO 027, you can see that that one is skipping about just as well as 147, while 043 is doing poorly. And this is still presented similarly in the patient cells. So while the skipping efficiency in patient cells is higher, the trends are similar. So they can be predictive of better skippability compared to another BMO targeting the same exon. So as a conclusion here for this part, a slow upstream intron, so 5' intron, leads to higher exon skipping efficiency if this is targeted with an AON. And then targeting in PMO closer to the 5' end of an exon has a significant higher chance of resulting in exon skipping. And here it doesn't matter whether the flanking intron is actually slow or fast. It's always relatively better to target close to the 5' end. So generating an out-of-frame transcript by exon skipping severely reduces observed efficiency, but cyclohexamide experiments, which inhibit nonsense mediate decay, but I will not be showing it due to time constraints, show that actually in our model system, the involvement of NMD for this is marginal. And then screening the potential for therapeutic exon skipping is underestimated in healthy control cells, but it's still useful to actually compare which AON is going to be the most efficient when comparing within the same exon. And intra-exonic differences in AON-induced skipping efficiency remain the same, whether you use a healthy control or a Duchenne cell line. Now, to actually look a bit deeper into what is going on with some of these non-sequential splicing events, we set up to make use of a BaseScope assay, which is a single molecule FISH assay where we make use of the fact that these signals can only be amplified when the probes, which the company calls Z probes, are close together. So for example, in an unspliced mRNA molecule, you'll have these Z probes far apart and you will observe no signal. Now the idea is if we use 2 different colors for 2 flanking intronic regions, so here intron 54 and 55, that we will see 2 signals if the transcript is mature. We will only see the first probe if the transcript is being spliced sequentially, but we'll only see the second probe signal if the transcript is spliced non-sequential. This is, of course, all a time-sensitive array, because at some point, every transcript will become a mature transcript, theoretically. This is still a work in progress, but what I can briefly share with you here is so here is a negative control of our muscle cells, and you can see no signal. Here are positive control cells, positive control probes for two abundantly expressed mRNA molecules, and you can see clearly the green and the red signal distinctly staining these cells. Now, if we look at our exon 44, 45 and 45, 46 binding probes, we can see, for example, that this cell here only has the 45, 46 signal positive, while this cell also has only has the green signal for the other molecule. And on the same side, we have this exon 54, 55, 56 probe set, and you can see that this cell has signals for both at the same time. Of course, this is quite hard to quantify. So what we've been doing is we have done some high throughput microscopy, and then by automatic segmentation, we are looking into whether the co-localization of the two probes in one cell nucleus, which is shown here on the y-axis, is different among different steps of differentiation. And I'm just showing you here that we're busy with this because it's a relatively finicky assay to set up, and we are very much still in the process of doing the analysis, but the probe specificity seems very promising. So we hope to get some good specific data for these events out of it. So that brings me to the final conclusion slide. So the Basescope duplex assay is viable in our cultured muscle cells for probing the dystrophin transcript. The designed DMD probes are seemingly functional, and but optimization of signal gain is necessary, especially when we are using fluorescent markers, because this is not actually supported by the company, but we are making do with it. And then the largest challenge is timing the differentiation of these cells right to catch the DMD transcript in the act of splicing our exons of interest. Because if you wait too long, you'll just have only mature transcripts. So it is a bit of a game of hide and seek with to catch these events, which start upon inducing the differentiation of the cells. And this project allows us to find new research topics to investigate the processing of the DMD transcript, such as the transcription rate, the splicing, and the stability in much more detail than we ever could before. Of course, I didn't do this all by myself, and just a quick excerpt. So from the LUMC Human Genetics, I would mainly like to thank Annemieke Aartsma-Rus for granting me the opportunity to work on these projects. Our technician Nisha Verwij, and then from the Leiden Genome Technology Center, Susan Kloot and Yafus, who've been indispensable for helping with the high throughput analysis of all the skipping data. And then, of course, Sarepta Therapeutics, the whole team has been very helpful and great to work with, but especially Fred Snell and Leslie, who have been very tightly involved in this project. And of course, I want to thank OTS and you for your attention.
Thank you so much, Remko. This has been great, very exciting data and exciting work. We'll wait until the questions come in the QA, but I have two quick questions for you. I think maybe you mentioned this, but what is the effect of having slow versus slow fast? Does the other side of like where your exon skipping has an effect, or as long as you have a slow in the beginning, a slow intron, then it's the same?
Yeah, so it's relatively the same. So that's exemplified here. So here we have the slow on the left for the 5' and then the 3' and slow fast, and it doesn't seem to matter much. Of course, there's quite some standard deviation, but you can hopefully appreciate that especially the 5' fasts are very hard to skip, even when looking at this one, which has slow, which is worse. Then as also like as an exception, you probably noticed these. This is exon 55, which is an outlier in our data set. And also one of the reasons why we were looking into this Bayescope assay, because we wanted to validate whether our hypothesis was wrong or whether maybe our assay was not sensitive enough to pick this up. That's why we're looking into so much detail into this region.
Yeah. Yeah, thank you so much. So 2 questions from the chat. Did you perform the Bayescope assay on muscle biopsies, or only you're validating it in cell culture for now?
That's a great question. Yes. So what we only did at the moment is we used control muscle cells. You could use this assay on paraffin fixed tissues, but these are probably, well, mature DMD molecules that we're going to find there. So because this assay for us hinges on the timing, but if you have other targets with Bayescope, you could definitely do it on biopsies.
Right. And a question from Margot: does slow fast or fast slow result in multi-exon skipping events as well?
It's actually very interesting because we know that quite some exons, if you skip them, they might drag along, so to say, another exon. And we expected these blocks that we saw. Let me just go back here. So we expected that these blocks would be skipped together, maybe. So if you skip exon 18, that exon 19 would come along. But this actually didn't happen a lot. We used different primer sets which were further away from the skip sites to check this, and we, even with high sensitivity assays, this effect was marginal, so, or not even detectable at all. So no, that we didn't observe that, but I do not want to rule out that it doesn't happen.
Right. Excellent. Another question just came in. So what is the timeline for capturing the exon skipping once the cells have differentiated?
I guess this refers to the Bayescope assay. Well, considering that the supposedly generation of the transcript takes 16 hours, we would say that if we are 16 hours in, we would have our first mature transcript, because all this splicing happens co-transcriptionally. So we really want to look into short differentiation times to actually see this 45 region or the 55 region. So yeah, I think in the ballpark of 8 to 12 hours.
All right. Thank you so much.
My pleasure.
Great. Well, thank you so much. We just want to wrap up the session today here with a few reminders. So we have a few webinars coming up. We have Art Craig on June 8th, Tim Yu on June 22nd, and Shalini Anderson on July 6th. So make sure you tune into those. They'll be great talks. I also want to draw your attention to the OTS mentorship program. It's a really wonderful experience to be able to be paired up and really kind of dive right into the field. The people, if you've had a chance to come to the conference, you'll know the people are great. It's wonderful to kind of just connect and be able to pull people aside and talk. But the mentorship program really offers more of this kind of one-on-one specific opportunity. And so if you have specific questions about that, you can reach out to Hassan. You can also email the website or the info@oligotherapeutics.org email address. And you can also find it at oligotherapeutics.org/mentor-program. I'll put that in the chat. So I just want to finish up by thanking everyone for attending, but especially thanking Remko and Priyanka for giving amazing talks today and sharing their work.
Thank you.
Thank you guys so much.
Great. Thanks, everyone. Take care.